Thank you all very much. I'm really very pleased to be here. I'm really pleased to talk in the Gatsby Plant Science Summer School. What I'm going to do is talk to you about a little bit about the International Year of Plant Health, but particularly I'm going to talk to you about why plant health matters. So why does it matter to us? And then I'm going to talk to you about some of the work we've been doing to try and combat plant disease, and tell you a little bit more about what we can try and do right now to try and make a difference. So this is the United Nations International Year of Plant Health. And of course, it was a year when we thought we'd be talking a lot about potential global pandemics caused on some of our major crop species. Of course, instead, we've got one that threatens our own species, and we've become slightly distracted by that. But nevertheless, we recognize the fact that if there was a pandemic affecting one of our major crop species, then it too would be an extremely serious issue for the world. So what I'm going to do is tell you, first of all, about, so why does plant health matter? How can we combat plant disease, and what can we do right now? So why does plant health matter? Well, plant health matters clearly because our food depends on plant life. It depends very much on plant life, and these are just some of the major crops that we grow around the world, and much of our land area is taken up in growing food, not only food for ourselves, but food for the animals that are then eaten by humans too. And despite the fact that there are around 30,000 or so edible plants, about 60% of the world's calories are provided by just 4 of them. So rice, of course, is a staple food for about half of the world's population. It provides about 23% of the world's calories, and it's consumed predominantly in Asia, where it's such an important staple. Wheat is by far and away the most traded crop that we have, around 1/4 of the world's calories provided by wheat. And it's a hugely important crop for our own consumption, but also for animal consumption too. Maize is really the fuel for much of the processed food industry. It's been estimated that an average US supermarket has got about 45,000 items in stock, and about 1/4 of those contain maize. So that means all of your soft drinks, beer, ketchup, coffee whitener, yogurt, cakes, snacks, all sorts of animal feed, toothpaste, nappies, charcoal briquettes, matches, batteries, all of those things have got maize within them. Even indeed supermarket shelves are often made of maize. The maize flour and the maize starch that's in tablets, in lining, gloves, all sorts of things contain maize. Potato too, of course, is a hugely important crop too. Interestingly, it's one which perhaps has some of the greatest genetic potential to be improved much further than has currently been carried out. But those 4 crops are vastly important, or hugely important to the world. When one looks at modern agricultural production, I think the thing that you're sort of struck by is just the genetic uniformity of crops, which is a result of extensive plant breeding. This is an Oxfordshire farm that I visited some time ago. And you can see just the incredible uniformity of the wheat crop across a field. This is a 3,200 hectare farm, about 12 square miles. And the other thing that always strikes you about this farm such as this is just how mechanized they are. And also the fact that they are very efficient in what they do. So the onboard computer map will look at the yield from last year, and it will talk then to the spraying devices, which will then ensure that nitrogen fertilizer is sprayed appropriately across the field according to last year's yield map. And the one here that one can see here has actually got chlorophyll meters, or it's actually got reflectance meters built into it, so it's doing fine scale adjustment of fertilizer across the crop at the same time. So incredibly efficient. So 12 square mile farm only employs about 2 people in this instance. So we're very efficient at growing food, and the UK can produce around 50% of its food. It probably could produce more, but we trade food very extensively too. But about 71% of the country's land area in the UK, employing only 1.5% of its workforce, contributing a relatively small amount of GDP, but a vast amount to our economic and, of course, health well-being. Despite the fact that agriculture, of course, is so efficient, there are lots of problems with contemporary agriculture. But arguably the most important problem that the world faces with regard to food production is still food insecurity. There are still 870 million people that are chronically undernourished, about 1 in 8 of humanity. Many of those live in low-income countries in the developing world. At the same time, we've got 1.4 billion people who are overweight. And we have huge inequalities within countries as well. So despite the fact that there's a huge obesity issue in the United States, for example, there are also about 11% of households, so about 14 million households, are also food insecure in 2018 in the US. And in the UK, indeed, there are about 3.6 million people who are food insecure, even in the UK. So food insecurity is a problem all over the world, but particularly in the developing world. And you can see that illustrated on this graph, where you can see that many of the countries which experience really severe food security are in Africa and South America and in Asia, but particularly in Africa. But of course, that's the situation now. In future, we have an even bigger hurdle that the world faces, and that's the fact that by 2050, we will have 9.7 billion people on the planet compared to the 7.8 billion or so that we have now. And we have to feed that growing population. The human population will then stabilize. By 2100, it begins to stabilize because the current annual growth rate is falling, as you can see in this graph. But that nevertheless presents us with a very severe problem, because we actually have to grow 60% more food by 2050. That's going to require 35% more fresh water, a lot more energy in a world where sustainability matters a lot more than previously, with less agricultural land because of the pressures of urbanization and food crops being taken out of production for biofuels. And all this in the face of climate change too. And to look at the scale of the problem, in Africa alone, we need a 260% increase in food production. So we have to produce more food than the world has produced in the last 10,000 years in the next 30 years. So this is a huge challenge for the world, one of a number that we face, but certainly a very, very big challenge nevertheless, and one that if you work in plant science in the future will absorb many of your professional lives. But we have grounds to be optimistic because we've sort of been here before to an extent. It's interesting to look back even just 100 years, so just after the First World War. This article appeared in the New York Times, and you can see there was widespread famine across Europe. There was widespread famine really stretching from the Balkans all the way through to Russia, and very severe food shortages and almost famine-like conditions throughout much of the rest of Europe. So we have had situations in the past where we've had really very, very severe food insecurity. What's happened, of course, is that there's been a huge increase in cereal yields. So there has been a 280% increase in global cereal production between 1961 and 2014. And that's actually kept pace. In fact, has been much larger than the sort of 136% increase in population, human population at the same time. There's been the twofold reasons for that. The first, of course, is that there's been very extensive plant breeding and crop improvement. We have much better high yielding crops than we had previously. And the other part is our ability to produce fertilizer. So we're still very heavily dependent on the Haber-Bosch process. Arguably about 3.5 billion of the current world population wouldn't be here if it wasn't for the Haber-Bosch process. So we can produce ammonia very cheaply from nitrogen and hydrogen gas, and we still make 500 million tons of fertilizer per year from that. But of course, that means that we're very heavily dependent on all of our agriculture on fossil fuel production. And of course, that is one of the big problems that we face, is that agriculture contributes 23% of our greenhouse gas emissions. We have some very unsustainable activity, particularly in terms of livestock production. As you can see from this graph, the carbon economics of producing beef, lamb, and so on are very, very poor. And although CO2 emissions from plant-based products are 10 to 50 times lower than animal-based products, they are still very heavily dependent on fossil fuel production. particularly in terms of fertilizer. So agricultural practices in the UK contribute 9% of our global of our greenhouse gas emissions, and around half of the sector's carbon emissions arise from fertilizer production. So we really actually do need to transform our entire agricultural system to increase resilience to that threat and dramatically decrease the carbon footprint. So what do we need to do? Well, we actually have some serious redesigning of major crop species to undertake in the next 30 years. This builds on the work that was carried out in the Green Revolution and the production of semi-dwarf isolates. So we saw a tenfold increase in the yields of rice during that period. So compared to the ancestral type of rice cultivars that we saw previously, to higher yielding cultivars starting with IR8 and other cultivars that were produced at the International Rice Research Institute. But we need to redesign them in future so that they have much better resource allocation than currently. So they're even higher yielding, but you can see by looking at the amount of root density that we need to generate, that they're much better at actually acquiring nutrients. So only in that way, by a very detailed understanding of plant physiology, plant molecular biology, resource allocation in plants, and plant architecture, will we be able to generate higher yields with lower inputs and greater resilience to environmental threats and greater efficiency. And that's very, very hard science. That's very tough science. It's going to engage a lot of us for many, many years. But there are some easier things that we could do. And what I'm going to tell you about is some of the easier things that we can do. Because I work on plant diseases, and plant diseases take every year between 20 and 40% yield losses. But this is a tractable problem. So we know that there is the technology emerging by which we can sustainably defend our crops from plant diseases. So each year, currently, plant diseases cost the global economy about $220 billion. Invasive insects, another $70 billion. Many of the world's most important diseases are caused by fungi, and we depend extremely heavily on fungicide application to grow many of our most popular crops. To give you a few examples, the current banana crop is threatened twofold. There's the perennial threat of black sigatoka disease caused by Pseudocercospora fijiensis, and then Panama disease caused by Fusarium oxysporum. Panama disease was responsible for wiping out the most commonly grown cultivar of banana, the Gros Michel, in the 1950s, and replacing it largely by Cavendish, the banana we currently grow. But that is itself very susceptible to the new tropical race 4 isolate of Panama disease, which really threatens production of Cavendish. Bananas take a huge amount of fungicide application every year to be grown. 30 to 40 times per growing season of triazole fungicides are sprayed to produce bananas. So it's not a very sustainable crop. It's one that the genetics of which are difficult, to plant breeding is difficult, and we really do require an urgent solution to Panama disease. Another disease of huge importance both historically and currently is coffee rust, which caused $1 billion worth of losses through coffee plantations in Central and Latin America in recent years. But of course, historically, it was discovered first reported in 1869 in Sri Lanka, and caused the collapse of the coffee industry in 1890 in Sri Lanka, where many of the producers switched to growing tea, and historians have credited this with the change from large-scale coffee drinking to tea drinking in the UK that took place. Another disease of huge historical importance, potato late blight, one that caused famine on these islands as recently as 1845, from which it's interesting that the Irish population has never really recovered. So the Irish population is still about 4 million, it's about 1 million lower than it was before the Irish potato famine. It caused a mass exodus of Irish citizens across the world, and this was caused by a combination of a terrible disease caused by an oomycete, not a fungus, an oomycete pathogen, Phytophthora infestans, and the consequence of the political decisions that were made as a consequence of that disease, which were very, very detrimental to the situation in Ireland. if all of those things weren't bad enough, historically and currently, the world's chocolate is threatened by threefold, by three different diseases: Phytophthora megakarya, also frosty pod, rot, and witch's broom disease. So three diseases which threaten cocoa and cause recent spikes in the price of chocolate. So these are all serious. Some of the serious threats that we currently have. So how do we combat plant diseases? So this has really been the motivation for the work that I do, the work that is carried out in my lab. And we study a disease called rice blast. Now, rice, as I've mentioned, is one of the world's most important crops. And we've been interested in trying to study rice blast in order to understand its really fascinating biology, but also think of ways in which we could potentially control the disease. This is an important disease. It's one that you can see from space. This is a Landsat image showing a great swathe of Southeast Asia being affected by rice blast disease. The disease manifests itself by these necrotic lesions on seedlings, which will coalesce and grow larger. They will grow up to 1 centimetre in length, and you get these large spreading lesions, and they'll produce between 20 and 50,000 spores every 24 hours. So there's a huge inoculum buildup, and those spores can then start new infections. The pathology that the farmers fear the most, though, is this disease called neck blast, which is where the fungus has spread now into the stem. This is a ring of sporulation around the stem of a rice plant. And under those conditions, it will rot away the inflorescence that carries rice grain, the panicle, and you'll get very, very severe yield losses. So when I spoke to the farmer when I took this picture, and he said that he had about 80% yield losses that year due to neck blast. The blast fungus, though, infects lots of other hosts too. So most recently, it's been affecting wheat. There's been serious wheat blast outbreaks. Wheat blast was confined to South America for many years. But it spread to Bangladesh in 2016, affecting 8 different districts of Bangladesh and destroying around 16% of the cultivated wheat area in Bangladesh. I visited Bangladesh last year, and I've never seen such widespread destruction caused by any plant disease. It was absolutely devastating, and it spread to 20 districts in 2019 and has re-emerged in 2020. So wheat blast is again a tremendously serious problem caused by the same fungus, Magnaportha oryzae. I became interested in Magnaportha not only because it was such an important global disease, but because it has some fascinating developmental biology. So the fungus starts the disease when 3-celled spores stick to the surface of a rice leaf. They send out a germ tube, which then develops into a special cell called an aprosorium, which develops pressure. The fungus physically breaks its way into the rice leaf and then will rapidly colonize rice tissue. And I'm going to tell you some of the biology as I talk you through the life cycle of the fungus. Here are those 3-celled spores. They carry with them their own glue. The glue was actually discovered by DuPont, who were trying to invent the mother of all Post-it notes by looking at discovering a glue that would stick very tightly to hydrophobic surfaces like Teflon. And it does stick the spores tenaciously to a leaf surface. They germinate, and then there's a fascinating morphogenetic pathway which leads to production of the infection cells. You can see that illustrated here. This is a 3-cell conidium. Each of the cells has got a single nucleus, you'll see in red. The nucleus in the cell that's germinating will divide. There it goes, formed by mitosis. And as soon as that happens, it triggers a pathway by which the other 3 nuclei that are left in the spore will die. You see there goes the first one, and you'll see the second and the third nuclei degrade. Because all the contents of that spore are degraded by programmed cell death and moved they're degraded by autophagy, a process of self-eating, and then they reassemble in the aprosorium where a huge amount of turgor is generated. So massive pressure is generated. And the whole process is linked to cell cycle control. That mitotic event is absolutely critical for infection to occur. The culmination of that is this cell, the aprosorium, this dome-shaped cell, which will stick tightly to the leaf surface and generate enormous pressure. So the pressure is 8 megapascals of pressure. So that's 40 times a car tire. So that's the highest pressure really recorded in a microbial cell. So enormous pressure. And it's generated by hydrostatic turgor. So a solute, glycerol, accumulates to very high concentrations and draws water into the cell by osmosis. That turgor is then translated into physical force at the base of the cell, at this structure called the aprosorium pore, where you see there's membrane curvature generation, and then a rigid peg ruptures the cuticle. And we've been interested in how that process is controlled. And we discovered it's controlled by, with the aid of some special proteins called septins, which are a type of, a part of the cytoskeleton. And they form this big donut-shaped assembly at the base of the aprosorium, which bounds the region where that pore is going to ultimately emerge and break the cuticle. And those proteins are involved in remodeling the cytoskeleton, which becomes oriented in a polar fashion, leading to ultimately to the repolarization of the cell. And if you delete any of those septins, then the fungus is rendered non-pathogenic. It can't cause disease anymore. That septin ring is found at the interface between the rice leaf and the fungal aprosorium. So it rigidifies the cortex of the cell and allows the fungus then to exert this huge pressure at the base of the cell. It's also involved in the organization of a whole set of other proteins at the base of the cell, which are polarity determinants. I won't name all of them, but there are a whole number of polarity determinants which are organized in that toroidal manner at the base of the aprosorium in the pore. And those are involved for the polymerization of actin. This is an actin-mediated protrusive force generation. And it only happens when the fungus has generated pressure. So once a fungus has generated a threshold of pressure, there's a sensor mechanism which involves a protein kinase. It senses that the cell has optimized its turgor, and then it will switch on all of this repolarization machinery and exert that force at the base of the cell. And we can find mutants that are unable to do that. So if we find a mutant that's unable to do that, this is a mutant, a turgor sensing mutant. This mutant, and we're looking at those same septins, which would normally form this big donut-shaped structure. And this cell will carry on developing lots and lots of pressure, but it will never exert that as physical force. And instead, that donut-shaped structure just collapses at the center of the cell, and the cell becomes very, very heavily melanized as it continues to build up pressure until ultimately it would explode, but it would never be able to penetrate a rice leaf. That molecular biology that we carried out has enabled us to carry out some mathematical modeling of aprosorium development. And we've come up with a model which suggests that this turgor sensing mechanism at the base of the aprosorium is responsible for an annulus or a toroidal structure of actin, of septins, which scaffold actin and lead to protrusive force generation and ultimately breaching of the rice cuticle. And that model is now something that we're using to guide further experimental evidence. So we can perturb our mathematical model and use that to guide further experiments by which we can study the spatiotemporal dynamics of actin polymerization, of cytoskeletal reorganization, to really understand how the cell works and what are the genetic determinants that allow it to work. Once the fungus actually gains entry to the cell, and now we're lifting the lid on a plant, some plant tissue, and we can see that rigid penetration hypha at the base of the aprosorium, the fungus then undergoes a different morphogenetic switch. So here we can show this in more detail. Here's the aprosorium again on the leaf surface. We can now computationally remove that rice tissue, and there's the fungus inside those rice cells. And you can see it's growing by this bulbous branched format. Those invasive hyphae propagate by something called pseudohyphal growth. They make buds and they grow into sort of in this phenotype, which you never see when the fungus is growing on an agar plate. All the time that's happening, the fungus is bounded by the plant plasma membrane. So it's occupying intact living cells. And it's doing that because it's continually secreting a whole battery of effector proteins into those cells. And there are two here. There's a green one and a red one. So there are two different pathways by which effector proteins are secreted. These are proteins produced by the fungus that end up suppressing plant immunity responses inside plant cells. And they're delivered either into the cytoplasm plants or into the apoplastic space. And there are many of them, and they're secreted in temporal expression patterns. Showing here this is the temporal analysis of around 500 different fungal proteins. And this is the expression pattern of their genes. Some of them come up very early. Those are expressed during the early stages of infection. Others come up during invasive growth. Some are specific to cell movement. Others during late invasive growth when plant cells are being killed. And others have fluctuating expression profiles. So there is very exquisite control temporally of gene expression by the fungus during its penetration of rice tissue. What that means is the fungus overwhelms the plant. It completely overwhelms the plant defense processes. And you can see that in this time-lapse movie. This is a 16-hour time-lapse movie of an intact plant on a microscope slide, and we've got the fungus fluorescently labeled with green fluorescent protein. And you can see how quickly the fungus will grow and ramify through all of those plant cells and fill the tissue completely over a period of about 16 hours. So it very rapidly overwhelms the host and grows very rapidly in those cells. And those are living cells that are being occupied continually. And it was really only by filming this structure, this process, that we were able to learn something about the way in which the fungus actually moves from one cell to the next. So you can see here's the fungus. It's found a junction between two rice cells, and it's swollen, and then it seems to undergo this hyphal constriction. There's an actomyosin ring formed here, and then it will squeeze its way through and then enter the adjacent cell. And by filming that structure, that process with live cell imaging, we were able to then study the biology of that cell-to-cell traversal mechanism. The first thing we did is we took one of these junction points and we did some electron microscopy, some high resolution analysis to find out what happens at those interfaces. So what I'm showing you next is a whole series of transmission EM slices, which I've assembled into a single movie. So what we'll do now, as you can see, here are lots of slices, EM slices through that transition point. And then we can tomographically build a 3D representation of the traversal point. And what you can see is the fungus will squeeze its way to around 300 nanometers in diameter, and as it moves from one cell to the next. And what it's actually doing there is locating pitfield sites, which are the sites where there are plasmodesmata, the cytoplasmic continuity between plant cells. And the fungus utilizes those to enable it to maintain membrane integrity of the plant as it moves from one cell to the next. And we've discovered a control mechanism for that process. So we made conditional mutants which are unable to carry out the cell-to-cell movement. This is actually a control experiment with one of those conditionally inactivated genes. And you can see that in the control, when we haven't inhibited this particular gene, we can see that the fungus is able to move very rapidly from cell to cell. So it fills those cells and then it breaks out through plasmodesmata into adjacent cells. And you can see there's almost like a quorum sensing mechanism where the fungus will actually fill epidermal cells, gets to a threshold of concentration, and then rapidly breaks out through those pitfield sites into the adjacent cells. If we chemically inactivate a single protein kinase, which controls this process, we add an inhibitor of that protein kinase to a mutant which can only be inhibited in this way with one single kinase, the fungus becomes completely trapped inside that cell. And that means we've got a mechanism that we can now study the biology of this cell-to-cell traversal mechanism. And what that's told us is that this is actually a specific morphogenetic process. And this is another stack where we've actually done some organelle tracking to actually study the structural biology of the cell-to-cell movement process by which the rice blast fungus magnaportha will move from one rice cell to the next. And this is a structure which we call the transpressorium. We didn't come up with that name. Someone had actually come up with that name in the 1950s by careful observation down a microscope. But what that suggests is that the fungus has a structure very similar to its apressorium on the leaf surface by which it moves from one rice cell to the next. And we can actually see the organization of organelles at those crossing points seems to be conserved. And you can see this really severe hyphal constriction as the fungus moves from one rice cell to the next. And that's enabled us to formulate a model for how the fungus is able to suppress plant immunity responses, host defense responses, at the same time as inducing cytoskeletal remodeling, formation of a septin collar, just like at the base of the apressorium, which enables the fungus then to move through those plasmodesmatal contiguous structures through whole pitfield sites, which they will find into adjacent rice cells. So this is the sort of fundamental biology of the rice blast fungus that we've been engaged in. But in doing that, this will lead us to long-term means to control the fungus, new ways of actually impeding its movement through rice cells and its ability to form disease symptoms. But what can we do right now? Because rice blast is an urgent problem. It's an urgent problem right now. Our lab is my lab's mainly concerned with fundamental plant, so cell and molecular biology, but we want to do something immediate. So we've been working on a project in sub-Saharan Africa where we've been trying to deploy a knowledge of pathogen population genetics to guide a sustainable rice breeding program. I'm going to tell you a little bit about that in the last few moments. So we know that plants can defend themselves against pathogens based on perception of those effector proteins that fungal pathogens produce. So normally fungal pathogens produce effectors which will suppress the initial plant immune response, the so-called PAMP-triggered immunity, by which they are able to recognize bacterial or fungal or oomycete pathogens. Effectors are produced, they suppress those plant immunity processes. Plants have countered that by producing normally intracellular immune receptors, which are the products of plant resistance genes. And they recognize some of those effectors, which are then called AVR effectors, and that leads to effector-triggered immunity or hypersensitive cell death. So we can see, for example, here a strain, this strain W0847, which is able to cause disease on a rice line which contains a resistance gene called PI50, because it doesn't have the effector that it recognizes, but it can't cause disease on PI2, PI9, or PIZT because it produces effectors that are recognized by those resistance gene products. Now, if we know that basic biology and then we monitor the prevailing rice blast population, we can predict which resistance genes would be the most valuable in protecting against rice blast disease in the field. And that's what we did. So we collected 1,000 rice blast isolates from right across Africa, so from north to south, so you can see, and from east to west, so both so from Mali in the north down to sort of Tanzania in the south, and going from both East and West Africa. We then genotyped them and we worked out which of their prevailing AVR genes, which effectors do they produce, and use that information to guide breeding. And we've actually sequenced many of these pathogen genomes as well. That gives us a matrix which looks like this, where what we can see is green is where we see resistance to disease and red is where we see disease. So we're just plotting rice blast cultivars versus blast isolates. By looking at the pattern, we can work out which of the resistance genes are able to exclude the majority of the rice blast population that's prevailing in Africa at the moment. And if we monitor that continually, we'll be able to have an idea of which are the most valuable resistance genes. Having done that, we were able to identify that there are 6 resistance genes which are very valuable in Africa: PI5, PISH, PI2, PI1, PI9, PIB. Any combination of those 6 would exclude quite a substantial proportion of the rice blast population of Africa, because the cognate AVR effectors are present in most of the isolates that we've identified in Africa. But we don't think that's enough. That's not going to be enough for sustainable, durable resistance. So we're pyramiding those with 3 other forms of resistance. That's a recessive form of resistance, PI21, and also 2 polygenic forms of resistance, different resistance specificities. And by pyramiding all of those different resistance specificities together, we believe we can have an ongoing process of durable rice blast resistance. But we also want to do that in cultivars which are grown widely in Africa, which are locally adapted to Africa, and which are popular with consumers. So what we're doing currently, the lines that we're focusing on are strains called Nerica 2 and Nerica 12. Those are both called, Nerica stands for New Rice for Africa. It's an interspecific hybrid between Asiatic and African rice. And Basmati 217 and Basmati 370, very popular in East Africa where they really like aromatic rice. And we have pyramided at the moment 2 major R genes in all of those, which are now being tested in the field. And we're pyramiding 3 different forms of resistance here, with which will ultimately be 2 major R genes and 2 a polygenic form of resistance and a recessive form of resistance. And those are currently in production. We then test those in hotspot tests across Africa. That hotspot testing, for example, is going on in Kenya currently, where we're looking at introgressions of PI9, and we're seeing how variable they are in effectiveness. both singly and in combination with other R genes. And we're also doing lots of training of young scientists to learn research methods in rice blast diagnostics as part of the project. This is a big project. We're looking at 19 different hotspots in 7 countries. So we're doing field testing in all of those different sites. At each of those sites, we carry out a multi-layered pathogen surveillance system. So we not only look for the prevalence of disease with those new isolates, but we continually monitor the prevailing rice blast population. We carry out greenhouse assessment in parallel with all of those strains, and we do laboratory diagnosis to look at the prevalence of specific AVR effectors. And all that information is built into a database in Nairobi as part of the project. The final part of the project is that we are carrying out plant clinics where we're training farmers and extension services professionals in rice blast diagnostics, and also telling them about the project and about what we're trying to do, how we're trying to use marker-assisted breeding to produce new lines of rice using cultivars which they know, they like, and they grow. And so far, we've trained 440 farmers in that project across Kenya. So far, we've got parallel studies going on in Tanzania and in Burkina Faso in the west. And that enables us to produce also fact sheets. So we produce a whole set of fact sheets that will go out to farmers, and online resources. There's an iPhone app that farmers are able to download, which helps them in rice blast diagnosis. It also provides information on the type of fungicide applications they can use if they have a current outbreak, but it tells them about genetic forms of resistance and the type of cultivars that are available and are being made available based on our knowledge of the prevailing rice blast population. So that project really has these 4 major outputs. We want to develop durable disease-resistant rice cultivars, an ongoing locally driven pathogen surveillance system, which is actually headquartered in Nairobi, which will carry on long after this project is finished and be run by local professionals, a network of trained professionals across Africa, and a very, very strong culture of farmer and consumer engagement by which we can hopefully win hearts and minds to the type of work that we're trying to do. And that addresses those 6 of the global development sustainability goals. So to summarize everything I've told you, and I know I've told you a lot in a very short period of time, ranging from cell and molecular biology through to population genetics. But why does plant health matter? Well, I hope that I've left you with information really about how we have this massive challenge. We've got to feed the human population to 2050. We've got to do that in a sustainable way. So sustainable crop disease control is really one of the first things that we can do, because sustainable yield increases and lowering inputs of crops is much, much harder scientifically to do, and represents, I think, one of the biggest challenges that we face. But if we can have sustainable crop disease control at an early stage, that will get us well on the way to the uplift in food production that we need. And what can you do? Well, I guess what can all of us do? I think one of the big messages that one sees, that the more that you hear and read about work in sustainability in agriculture, is an increasingly plant-based diet. I think has to be a sort of lesson for us all, really, in terms of our sort of responsibilities to the planet. How can we combat plant disease? Well, I think a fundamental understanding of cell and molecular biology is vital. We need to understand microbial pathogenesis in detail. It's a very elaborate, orchestrated, highly regulated mechanism, which involves intricately linked to metabolic control, cell cycle control, and environmental signaling. We also need the same level of understanding of plant immunity. Only in that way can we identify durable targets for intervention and predictive manipulation of plant immunity, which is really what we're trying to do. Is if we understand plant immunity in real detail, we can predictively manipulate it so we can have durability against any crops. And that's something the Sainsbury Laboratory is really committed to doing. And what can we do right now? Well, we can certainly deploy resistance gene combinations. We know we can do that. We don't even need genetic modification to do it. We can do it through marker-assisted breeding and selection, pyramiding of genes. It's easier by GM. It's much easier by genome editing, where we're able to do that. In that way, we can reduce fungicide use dramatically, very dramatically, and build towards sustainability. We can also work immediately in low-income countries to generate durable resistance to emerging plant diseases. But we can only do that if we build local capacity and expertise, and really harness the local enthusiasm and expertise that's available in order that those processes themselves are sustainable and they're not just associated with projects of the type that we're currently leading. And what can we all do? Well, we can all be more responsible consumers, because I think only by doing that can we exert the type of consumer-led political pressure that we need to if we are to change agricultural systems across the world. So I'll leave you with those thoughts. The people who have helped, I won't be able to mention all of them because there are just far too many, but I'm very lucky that I've got a fantastic group of people in my lab, PhD students and postdocs from all over the world. They're a wonderful group, and I feel always very grateful for the work that they do and the dedication they show. I equally have fantastic colleagues at the Sainsbury Laboratory. My colleagues are great friends and foils to the work we do. They provide challenge and intellectual rigor to the type of work that we're trying to do. The same is true across the whole of Norwich Research Park. I have also had great colleagues in my former home at the University of Exeter, and these great collaborators again across the world, and the funding agencies, particularly Gatsby, who fund the work at the Sainsbury Lab, but also the work we've done, I've talked about in Africa, funded by the Gates Foundation, GCRF and BBSRC. And the Helping Scholars Programme has funded 8 PhD students so far from the developing world, from rice-growing regions of the world, to come to my lab and train in rice blast research. Okay, I'm happy in the Q&A to answer any questions that you've got, so thank you very much.